Hi, I'm Dr. Christopher Bunick, Associate Professor of Dermatology at the Yale School of Medicine. I'm here in New York City at the Summer AAD Innovation Academy, and I gave a talk in the Pipeline Innovation Therapy session specifically focused on the future of small molecules in dermatology. In this talk, I took a unique approach. I didn't go through all the clinical trial data of the current small molecule therapies. What I wanted to teach were the different concepts that go into designing and developing a small molecule drug and how that's going to impact how the specialty thinks about new drugs that are developed in the future. I started by focusing on 4 key pillars or areas that you have to think about when you design a small molecule drug. The first being the mechanism of action that you're targeting, the precision chemistry with which you develop the drug, third, the exposure in the tissues, the way and at the level you want, and then finally, how all that connects to impact the patient care. Some examples that I gave. I used the JAK inhibitor family to talk about how we think about mechanisms in terms of selectivity and tissue targeting. For example, there's a new dual-acting JAK1 TYK2 inhibitor, brepocitinib, that's on the verge of approval for dermatomyositis. I talked about how this drug captures in one molecule multiple pathways critical to that particular disease. From that, I used it as a springboard to talk about immune pathways in dermatology and how the overlapping nature of Th1, Th2, and Th17 pathways necessitate thinking about how to target specific pathways with small molecules. In the precision chemistry component of my talk, I used a number of examples to highlight concepts that we have to think about. For example, using PDE4 inhibitors as a great example, I talked about how potency differences can occur as we learn more about how current drugs work. We can develop more potent drugs. But beyond potency, you also have to understand a few other key components, such as, well, how does the molecule actually bind its target? And I used a primolast as an example where a primolast has a 90-degree kink in the molecule that affects its potency, but also the side effect profile with regards to its central nervous system penetration. I then highlighted how metabolic pathways are really important in thinking about the design of drugs. In particular, roflumilast has a metabolite known as roflumilast N-oxide, and a new drug being developed, erismilast, is very similar to this roflumilast N-oxide. So this is a great way to highlight that understanding the metabolic pathways and how these drugs get broken down in the body could be clues to being able to harness a specific metabolite and use that as a new therapy in dermatology. I then went on to talk about STAT6 degraders and the degrader small molecule family as a whole. This is an emerging new family in dermatology and highlighting the fact that targeted degradation can be a very effective therapeutic strategy. In addition to that, I talked about covalent small molecule inhibitors. We have 2 in dermatology right now, ritlecitinib and remibrutinib, that both use acrylamide warheads to covalently bind cysteine residues in the target. And ultimately, both these molecules have activity within the TEC kinase family and do it in a very covalent, potent way so that you can treat, respectively, alopecia areata or chronic spontaneous urticaria. And then I highlighted a couple examples of my own patients with these drugs to really connect from bench to bedside, that translation of how the chemistry of the small molecules actually matters in terms of taking care of the disease pathophysiology in the patient. And I concluded my talk about talking about exposure. And there's no better example than oral minoxidil to learn about how exposure and manipulating and controlling and engineering delivery systems that control exposure than minoxidil. Minoxidil is not an active drug. Minoxidil sulfate is the active drug. And there's an enzyme, SULT1A1, a sulfotransferase, that has to sulfate minoxidil. But what happens with immediate release minoxidil is that for about 2 hours, that minoxidil floods the system and the enzyme can't process all of that. Most of that minoxidil goes to waste. But if you develop an extended release formulation of that minoxidil, now that enzyme can handle all the minoxidil coming its way and make more minoxidil sulfate over, say, 8 to 10 hours as opposed to 1 to 2 hours, showing that you don't have to, in order to improve the pipeline in dermatology, necessarily invent a whole new drug. You can take an existing drug and just actually deliver it in a better way so that you can not overwhelm the enzyme needed to activate that particular drug. So ultimately, in my talk about the future of small molecules in dermatology, I emphasize a number of these concepts. When you see a new drug or you're thinking of developing a new drug, you want to think about all these concepts and how is this drug addressing all these concepts to help our patients.